all for coming today. This morning, I'll be discussing a paper titled "A Critical Role for the Cannabinoid CB1 Receptors in Alcohol Dependence and Stress-Stimulated Ethanol Drinking." Or, in simpler terms, drinking habits of receptor-deficient mice under stress. So why should you care? So, this paper relates to the greater problem of addiction. Now, as you can see by the statistics on the slide, addiction is a problem that affects many people within our own country. 120 million of people over the age of 12 were current users of alcohol. Of those users, 18 million met the criteria for substance abuse or dependence on alcohol. Before we can really delve into this paper, we have to talk about stress for a moment. So, what can our stress make us do? Imagine if you have 2 tests and 2 quizzes.
Oh, God, please, no! 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 No!
Wow, okay. Raise your hand if you have ever had to exercise to clear your head because of stress. Raise your hand if you have ever had to stay up late doing work because of stress. Raise your hand if you have ever had to skip a meal because of stress. Now, answer only in your head. Have you ever thought about cheating on tests because of stress? How about turning to drugs or alcohol because of stress? We consider actions such as the first few as I listed as more or less normal, or rather less detrimental to our own health. But what we don't consider is that stress is an extremely powerful motivator. Therefore, it's important to study how stress can influence our lives and behaviors in such profound dimensions. So let's talk about addiction for a moment. What is addiction? Addiction is a disease that is characterized by a few key hallmarks or principal traits, one of which is relapse. So relapse comes in 3 different forms. First, you have drug-based. Pretty self-explanatory, drug-based is simply the re-exposure of an addictive substance that reinstates drug-seeking behavior. For example, if I'm addicted to cocaine and I stop doing cocaine, but I have a teensy little bit of cocaine, that will reinstate my drug-seeking behavior. Second, you have cue-based. Now, some of you may know the famous behavioral scientist Pavlov and his renowned conditioning experiment with the meat powder, the bell, and the dog. But when I talk about cue-based relapse, I have my own story of the fabled brown chair. So let's say every Sunday morning, as the sun begins to rise, I sit in my favorite brown chair and I do heroin. Now eventually, I begin to associate these contextual cues, such as the brown chair or the rising sun, with receiving the drug. This process is a fancy term called long-term potentiation, but that doesn't matter. What matters, though, is that with enough repetitions, if I were to stop doing the drug, but if I were to be placed back with those environmental cues, such as sitting in the brown chair or seeing the rising sun, that could reinstate my drug-seeking behavior. Now lastly, you have stress-based relapse. Once again, pretty self-explanatory, stress can lead to the reinstatement of drug-seeking behavior. For example, if I'm addicted to meth and I stop doing meth, but I'm super duper stressed, that can lead me to seek meth once again. Now, interestingly enough, stress is also involved in initial drug-seeking behavior. The pathway that relates to addiction is called the endocannabinoid pathway. This paper focuses specifically on an integral receptor within that pathway called cannabinoid receptor 1, or CB1. A receptor is merely a protein that's on the surface of a cell that can tell that cell or other cells to do something through messaging called signaling. They're very important for the functions of our body. So this paper specifically focuses on how that receptor, CB1, is involved in stress-based ethanol drinking within mice. So now we get to the experiment. So the researchers had 2 cohorts of mice. The first we can refer to as the wild type. They're what we would call normal mice. They have CB1. Whereas the second group had a genetic mutation that resulted in a complete deficiency of CB1 because the gene that encodes for CB1, called CNR1, was silenced. To recap, 2 groups. One with CB1, the wild type, and one without CB1, the knockout. So first, the researchers wanted to see if the presence of CB1, or the lack thereof, would affect behavioral responses to ethanol in both the mice groups. So they ran, logically, a wide variety of behavioral tests after administering ethanol, 2 of which I've included on the slide. On the left, we're looking at hypothermia induced by ethanol administration through an intraperitoneal injection, an injection through the chest. On the right, we're looking at locomotor function. Now, as expected, the researchers established a dose-dependent correlation between the increasing amounts of ethanol being administered along the x-axis and the intensification of the behavioral responses. So, for example, as you administered more ethanol, the mice had a greater change in body temperature. The white bars represent the wild type, whereas the black bars, they represent the knockout. But that's not what we need to take away. What we need to take away is that the presence of CB1 or the lack of CB1 did not affect the behavioral responses to the ethanol administration. Can anyone raise his or her hand and explain back to me what I just said in their own words? I will wait. Yeah.
Okay, so you had 2 groups. One was wild type and one was knockout. The wild type had the CB1 receptor. They then did tests on both sets of group behaviorally to see what happens when you administer ethanol. One had to do with body temperature and one had to do with locomotor control. They found that with the body temperature, as you administer more ethanol, the body temperature change increased in both the knockout and the wild type groups, showing that there wasn't any difference in this way.
Yeah, great. That was super detailed. That was awesome. So, moving on. For the purpose of a later part of the experiment that the researchers wanted to conduct, they first had to run a 5 or so week conditioning regimen. The purpose of this regimen was to condition the mice to drinking ethanol. So the setup was that each mouse had in its respective cage a bottle of 8% ethanol solution and a bottle of water. Now, interestingly enough, the researchers observed that the mice without CB1, the knockout, as indicated by the trend lines with the circular data points, had an initial preference for ethanol, as compared to the mice with CB1, the wild type, indicated by the trend lines with this triangular data points. Now, on the left, they're looking at relative preference ratio from one group to the other, whereas on the right, they're actually looking at the quantity of ethanol consumed. But as you can see, the difference subsides around week 2, leading the researchers to say that the presence of CB1 or the lack of CB1 will not result in any proclivity towards ethanol receiving. Moving on. So, this is what really lies at the crux of this paper. After doing that 5 or so week conditioning regimen that we just looked at, the researchers decided to administer a stressor in the form of a foot shock to the mice. They measured before, 24 hours after, and 96 hours after the same metrics of relative preference as well as ethanol, absolute quantity of ethanol consumed. On the left, preference; on the right, consumption. So what they saw, though, interestingly, well, as expected, because we established that stress can lead to drug-seeking behavior as well as relapse, that the mice with CB1, as indicated by the bars with the positive, had an acute spike in preference and consumption that quickly subsided by 96 hours after to the pre-stressor-induced levels. But the researchers noticed that in the mice that didn't have CB1 in the knockout, there was no change in preference, implicating CB1 in stress-based ethanol drinking within mice. Now, just by looking at the graphs, can someone explain to me why we can deduce that CB1 is involved in this process? Yeah.
CB1 is present, the process doesn't happen.
Yeah. Great. So this raised the question of maybe CB1 affects how stress was exhibited or transmitted within the mice, or rather, if the mice were stressed the same. So what they looked at first were behavioral responses to the stressor. So what they measured were jump responses that were observed after the foot shock was administered to both groups. On the left, again, we're looking at the wild type, whereas on the right, we're looking at the knockout. And what they saw, though, was that they had similar behavioral responses to the stress, leading the researchers to believe that they were stressed the same. But this is behavioral neuroscience. You can't make claims like these solely off of a behavioral response. So they had to look at the brain, which is what they did next. So the researchers looked at neuronal activity, or activity within the brain. A neuron is just a fancy name for a brain cell. Now, what they were measuring were something called CFOGs. which is merely a biomarker that is released when neurons fire. Neurons firing just means brain cells activated. So by looking downstream at CFOS, what's expressed, they can see roughly the activity within the neurons. Now, they looked at the brains of both the wild type and the knockout that had received the stressor, the foot shock, and that had not, in order to have frames of reference for comparison. So as you can see, on the second and fourth columns, you're looking at the wild type and the knockout that had received the stressor. And they saw that they had roughly the same CFOS expression, leading them to believe that they had roughly the same neuronal activity. So moving on, let's talk about what we've learned thus far. So first, we saw that the presence of CB1 or the lack of CB1 did not affect the behavioral responses to the ethanol. Second, we saw that stress, as expected, induced an acute spike in preference and consumption in the wild type, but didn't have any change in preference or consumption in the knockout. But what we implicating CB1 in stress-based ethanol drinking within mice. But what we just saw was that CB1 did not affect how stressed the mice were or the general neuronal transmissions. So how does CB1 fit into all of this? So this paper, it's fairly old, but it's quintessential. Published in 2003, it laid the foundation for a lot of research that has come since. Now, I've included on the slide a few papers that are really important, a few of the many that have been published on CB1 since then. Now, essentially, we're not going to go into detail about these papers, but what is sort of established is a lot of conflicting information. For example, some papers will say through their experiments that CB1 is just involved in learning, not involved in stress. And some others say the converse, that CB1 is just involved in stress, but not involved in learning. Now, let's talk about some of the paradigms that they used. So, you start with, as we talked about Pavlov in his conditioning experiment, we have to talk about learning a little bit. So first, you have a cue. It's not really a cue it's called a conditioned stimulus. So that would be the bell. Then that's involved in two types of associative learning. You can have appetitive, which merely means pleasure-based. So that would be the equivalent of the meat powder associating with the bell. So that would be with pleasure. Or you can have a conditioned stimulus, such as a red light with a shock, which would be what's called aversive learning. Now, moving on this leads us to our future research. CB1 needs to be studied independently, as in isolating learning while focusing on stress, and vice versa. Now, in terms of what they actually need to do specifically, they need to look with receptor antagonists, something that would knock CB1 down, such as AM251 or rimonabant, as well as with agonists, something that would promote CB1, such as THC or WIT. By researching with an agonist, something that would promote it, an antagonist, something that would knock it down, as well as with controls, you'd be able to have fuller results in order to study CB1 better. This is what my IRT project is planning to do for this year. For our appetitive learning protocol, we're planning to use something called a blue dot pleasure chamber. This purely means that the zebrafish will associate the blue dots within the blue dot pleasure chamber with receiving either an aversive stimulus, such as a shock, or an appetitive stimulus, such as food. Therefore, we believe that this blue dot pleasure chamber will be a successful model for us to use. So, hopefully, we'll have some success going forward. Lastly, I'd like to thank these people for helping me not only get my presentation to where it is today, but facilitating my growth as a scientist in my general science career. Thank you all for coming so much. Hope you took something away.
Questions, comments, concerns, emotions?
Can you go back to the slide that had there are a bunch of different article titles that people worked on? See this one? Okay. Did any scientist ever think that CB1 receptor might not play a role in acquisition of fear aversive learning, or that Lutz group just said for sure that it might?
So you're asking if.
Because you said there were a lot of different ideas as to what this receptor does in the whole situation, and that one just says that it might play a role. But did anybody have any differing ideas, or is that sort of like the furthest they went from that sort of?
Yeah, so it's been, before this paper, it was implicated in stress and learning, but a lot of papers that have come since this have said exclusively that it's involved in stress or learning and contradicting each other. So there's a lot of misinformation, a lot of lack of clarity as to what its role really is in terms of the pathways. Yeah.
Do you know how they looked at the grades?
As in, like, what did they do?
Yeah.
No, I'm not sure specifically like what they did in order to procure these results.
Have there been any more studies that established what the role of marijuana can play in all this? Because you mentioned at the end that THC is an antagonist for CG1, and that's, it's not just a brain protein, that's something that's like a lot of people have access to.
Yeah. I can't think of some off the top of my head, but I can definitely look into some and get back to you. Good question, yeah. Yeah.
Do you think that the addictive nature of the drug that was being tested would have an effect on the overall study? Like whether the drug was someone was more prone to becoming addicted to it, like say nicotine or a different drug, you know what I mean?
Could you say it again? Sorry.
Do you think that the nature of the drug that was being tested, whether like if somebody had a higher likelihood of becoming addicted to the drug, would that affect the receptor?
Yeah, so actually, interestingly enough, there are some, groups of populations of people that have what's known as alleles that, there's no concrete studies saying that, some people are more likely of being addicted, but there have been specific groups that look at and see that certain populations of people, have a mutation that affects how they, process alcohol, how that could be, I don't know if it can be extrapolated to if they'll likely become addicted, but I can get back to you, sorry. Yeah.
You mentioned that the thing for the receptor might also be involved with learning in general, not just stress. Do you know if the researchers, any of the researchers past this, had any thoughts about whether removing it would have other adverse side effects?
Would have what side effects?
Would have other adverse side effects with regards to learning in general?
I'm not sure. Sorry.